Good afternoon and welcome to this Robinson College webinar alumni forum. I'm Judith Richards. I'm a senior member of the college. I'm a retired consultant medical microbiologist and director of infection prevention and control, and I was the past chair of the International Federation of Infection Control. As some of you may know, especially those of you who've had the opportunity of seeing the video recorded by Steve Trudgill, who's the president of the Alumni Association, we were due to have this meeting in March, but unfortunately we were overtaken by events, had to wait a long time, and hence this webinar. However, this has also given us the opportunity to link up with the university's Alumni Festival, which is great because we have been able to open up the meeting to many more participants. So a warm welcome to you all. I would like to thank the college's development office and specifically Sarah Westwood and Norbert Trusinski, not only for helping us to set up this webinar, but also guiding us through the required knowledge and actions to make it work. I've got a couple of very brief announcements. You'll be pleased to know that I'm not going to be talking about fire exits and what to do in the case of a fire alarm. But for those of you who are newish to Zoom, please note that all participants, except for the 3 panelists, will be muted throughout the presentations. There will be opportunities for for asking questions, and we'll have a discussion towards the end. And if you've got any questions, please post them by using the Q&A button at the bottom of your screen, not the chat one, although you can chat amongst yourselves. But for any questions to the panelists, I would appreciate if you could use just the Q&A button. And also, if you've got any questions specific for any of the panelists, please indicate the name of the person that you're asking the question from so that we know who to address it to. Otherwise, they'll go in the general pool. Given the time that we've got, I do apologise if we're not able to address all the questions that might be posted. All 3 participants will give us a brief overview of their life in college, will talk to us about their careers and how they have developed, what they've been doing over the last few months, I hope, and how they see the big title of the Future of Medicine, which is a huge ask, but I'm sure that they'll be up to the challenge. So I'll introduce very briefly our 3 panelists in alphabetical order. Professor Wendy Barclay, she's the head of the Infectious Diseases Department, virologist at Imperial, and her main area of interest has been for a long time respiratory viruses, especially cold and influenza viruses. So it's become very topical, and I'm sure she'll touch on a few topical issues there. Professor Adam Fox at Guy's and St Thomas's, he's a professor in pediatrics, and he's the commercial medical director at Guy's and St Thomas's for his sins, and he'll tell us all about that. And he's got a special interest in pediatric allergy. And finally, Professor John O'Brien, he's an old age psychiatrist, national lead for dementia research, and he's at Cambridge Department of Psychiatry and Neurosciences. So I'll hand you over to our first speaker. Wendy, welcome, and thank you very much for making the time to be with us.
Have I unmuted? Hello? Can you hear me, Judith? That's not a good start, is it? I think I'm still muted.
We can hear you, Wendy.
You can. Thank you very much. That's a relief. Okay, so thank you very much to Judith for that introduction. And it's a real pleasure to be here. I think all 3 of the panelists are looking forward to sharing a few memories from Robinson College. My time spanned from 1982 to 1985. And when I look back on college, it seems to be one endless sort of string of parties and balls and friends. I'm afraid I don't remember very much about studying. I remember that I made some wonderful friends and was very happy. I am grateful to Mary Stewart, who was my tutor at the time. And I'm grateful to all the friends and all the opportunities that I had there. I had quite an active social life outside of college, because I came to college a very keen dinghy sailor and was lucky enough to get into the Cambridge University team and sailed for the university for 3 years. So I had a super bunch of friends and used to drive them around. It was one of the only excuses to be able to have a car in college at the time. So that was quite a luxury. I was also a member, not a founding member, but a sort of fairly old member of the Robinson College Girl Fridays, which was a ladies' society who had the motto for their parties of black tie and no trousers, which were always lots of fun. And as I said, I remember I made some fantastic friends. I remember long summer days, lots of garden parties and croquet, and sitting after exams were over on the grassy banks at the back of college. And despite all that partying, I did make it through to the end and come away with a view to thinking that I really did enjoy the science that I'd learned there and wanted to make a career of looking in more detail at biological sciences. And I was lucky enough to secure myself a place to do a PhD, but it was a very unusual PhD. Back in the day, there was the opportunity, it was rather like a case award. So it was partly with industry, partly with a university, and partly with a very infamous place now called the Common Cold Unit, which is pictured here in this black and white photo. And this was a place that lived on the top of Salisbury Plain. And it was possible for people from all over the country to come here for 10 days holiday for free. In fact, they even got a little bit of pocket money, as it says there in the advert. And the idea was that we would infect them with a mild cold and then study them. And my job as my PhD was to try to find out whether or not people became immune to the colds that they'd been given. And I had a trusty band of volunteers who came back the following year and allowed me to try to give them the same cold again. By measuring the antibodies, I was trying to test whether or not they had become immune and whether or not the correlative immunity was serum or nasal antibody. What was really fantastic about this opportunity was, of course, it's quite a clinical virology style of project and got me interacting with a fantastic bunch of clinicians who run the Common Cold Unit, which was headed at the time by David Tyrrell, a really famous virologist. But actually, the year I began that PhD was the year that the first ever three-dimensional structure of a virus was published. And that's this beautiful picture you see on the right-hand side. That is human rhinovirus type 14. And X-ray crystallography had finally managed to solve such a structure. And the industrial part of my PhD was really to see whether or not one could use knowledge from this kind of modern day crystallography to make vaccines that would perhaps prevent against all colds and not just the one serotype that we were interested in. So it was a fantastic experience, and it taught me everything from sort of molecular approaches right through to clinical virology. And after that, I was lucky enough to get a couple of postdoctoral positions. One was in the United States at Mount Sinai Medical Center. And the one Christmas I spent there, of course, the skyline was such that one could still look out and see the Twin Towers. And my subject there was influenza viruses, so no longer rhinoviruses, but influenza viruses, another important respiratory viral infection of humans. And this was at a time when my mentor, Peter Palese, there had worked out how to perform what we call reverse genetics with this class of viruses. And that's the ability to create viruses de novo from pieces of DNA. This is one of the things that really had fascinated me when I turned to virology as my subject. Viruses are so simple that you can actually synthesize them in the laboratory. And by doing so, you can pick them apart and understand what each little bit does, what every gene of the virus is for. So we applied that reverse genetics technique in Peter Palese's laboratory to try and understand rhinoviruses and influenza viruses, and also to think about how one could make the next generation of flu vaccines, for example, and indeed the live attenuated flu vaccine of today is created using this reverse genetics technique. And really, influenza virus for me has been a fantastic subject to focus my career around. I returned in 1992 from the United States and set up my own group to study influenza. And the thing that's great about influenza is that everybody understands the importance of it. And you can come at it from so many different angles. One can think about how to develop better drugs to treat the virus, better vaccines, and also that there is a constant threat, as I'm sure everyone's well aware nowadays, of pandemics of respiratory viruses. And usually people think about influenza as the sort of prototypic virus that crosses from animals into humans and sparks a new pandemic. And certainly during the time that when I was sort of initiating my own group and stepping forward in my career, the threat of bird flu, which arrived in the late 1990s and sort of hung over us for the following decade or so, was huge and was a good sort of starting point to write grant applications and focus minds. In 2007, I moved from Reading University, which had hosted my first sort of independent part of my career, to Imperial College London. And that brought me, of course, into close proximity with many fantastic colleagues that Imperial offers, but also with the media, who are very much sort of based in London in terms of studios. And two years later, in 2009, the world experienced the first respiratory virus pandemic of the 21st century, which was swine flu, or H1N1 2009. And I found myself as a sort of expert in that area, being called upon to speak often to the media, really bringing home to me the importance of science communication. And I think that's something that stayed with me very much and has served me well in recent months. So turning to recent months, what has the COVID experience been like for me? If I sum it up in one word, I think busy is the word. As a person who's worked for the last couple of decades in respiratory virus infections and thinking about pandemics, my time has been drawn upon, and I sit on a number of advisory bodies, trying to see the way through and give scientific-based advice to help difficult decisions be made. Over the past 6 months, some of the areas that I've been asked to focus on particularly are to think about things like viral load. How much virus does a person carry? How do we measure that? And what does that mean? Do people with more virus in their body, is that the reason that some people fare badly or not? How long does a person remain infectious to others for? Can we refine the assays that we use when we diagnose people to be able to release people perhaps from isolation earlier and to understand the risks of sending people home from hospital back into somewhere like a care home and whether or not they might be onwardly infectious to others? How is the virus transmitting? Is it airborne? Is it through contact, etc.? And will the virus evolve? These viruses that I've worked on my entire career are RNA viruses. They're inherently genetically unstable and have this amazing capacity to change. As more and more of us become infected by this new virus, SARS-CoV-2, one might expect the virus to begin to adapt to its new human host and also change its appearance in some way, which may have extremely important implications for the way we control it moving forwards. At Imperial, interacting with some of my colleagues, I've been very much involved in huge studies, a program called REACT, in which we send out home tests to 150,000 people at a time to try to measure the prevalence at any one time in the United Kingdom, and also antibody tests that people can perform in their own homes to measure seroprevalence as well, to get a feeling for how much immunity we have at the moment. And that brings me to the other sort of pressing question which we're trying to address, which is about waning immunity. And this brings me right back to my PhD. I performed my PhD studies on a rhinovirus, a common cold virus. And as I told you, I infected volunteers with that rhinovirus and invited them back a year later. At that time, a year later, they still had antibodies to the virus I'd given them a year ago. And when I tried to infect them with the same virus, they were immune. Those antibodies were protecting them. Another lady who worked at the Common Cold Unit at the same time as me did exactly the same experiment with human coronavirus, not the new one, not SARS-CoV-2, but one of the seasonal coronaviruses, which causes around 20 or 30% of common colds every year. And her results were completely different. Her volunteers came back a year later and their antibody had all gone away. And when she challenged them again with their coronavirus, the same coronavirus, they became infected. So this issue of waning immunity comes right back to the experiments that I was doing 25 or 35 years ago. It's been a tremendous time. The final part of my task today is to think about the future and what are we learning now that may set us up for a better response next time, because there will be, I'm sure, another respiratory virus which crosses from animals and causes another human pandemic. I'm sure everybody will be familiar with the term moonshot at the moment in the media. At the moment, it's very much used to describe the concept of rapid tests, perhaps home tests, that will help testing capacity. But in my view, the moonshot is also about another project, which is also called Moonshot, by the way, which is a bunch of very clever structural biologists and chemists who are trying to design new drugs that will directly act against this new coronavirus, SARS-CoV-2. And I think if we were to be able to combine those two aspects of the Moonshot program, and people in their own home could see that they were infected and immediately begin taking a directly acting antiviral, we might be in a much better position to control this virus, because the problem with viruses like flu, rhinovirus, coronavirus is that they are acute infections, and by the time you can begin to treat them, probably the transmission has already occurred. The other thing I'll mention briefly is SARS-CoV-2 vaccines. What do I think the future will bring? You may or may not be aware that there are more than 200 SARS-CoV-2 vaccines in some part of development. The one that colleagues of mine at Imperial College are trying to develop is a self-amplifying RNA. The idea of this is that you inject the RNA in and your muscles do the work of being the vaccine factory. So no more need for huge vaccine production plants, but this could become a much more distributed and localized vaccine production proposition where countries, even low-income countries, could be in control of their own vaccines. And this is a very adaptable system so that every new pandemic that comes along, you could have a vaccine to meet the new need. And my own research, the sort of pinnacle of my career, I suppose, came a couple of years ago, where I very proudly discovered a particular protein, which is extremely important for the influenza virus. And this is a protein called ANP32, and we now even know the crystal structure of that protein and the way it interacts with the polymerase enzyme of the virus, shown here in the top left-hand corner. And we're currently pursuing what some people might think is quite an outlandish idea of a new way of thinking about how to prevent future influenza pandemics by using CRISPR genetic change to alter the genome of domestic poultry, such as chickens, which we know are a way by which these viruses can cross from wild animals through domesticated animals and into humans. And the concept here is that we might be able to build chickens that are altered in very precise ways, which means they are resilient to influenza infections and can no longer be that conduit into man. And I was really privileged about 2 years ago to be able to sort of pitch this idea at the World Economic Forum in Davos to a number of sort of interested parties. So I'm going to finish there. I hope that I have sort of given you a sense of the journey I've been on since leaving Robinson. It's been a fantastic time. I couldn't have done it without my wonderful husband, children, my father, my family who've supported me all the way through, and also the other family, the family of young people whose PhDs and postdoctoral careers are spent at least some part of time in my lab. And yeah, thank you very much for listening to me.
Thank you. So shall we move on then? Adam?
Thanks very much, Wendy, giving me quite something to follow. Going to share my screen. Lovely. Okay. Can you all see and hear me okay? Brilliant. Okay. So I was also asked to think and reflect on my Cambridge journey. And actually I can recall exactly where that started. It was in the school library with 2 or 3 friends when we were very carefully going through the back of the prospectus, the paper prospectus for Cambridge, because at the back, some of you may remember this, it would list the number of places and the number of applicants to each college. So we thought we'd be clever and we worked out all the ratios. So we worked out exactly which college would give you the best chance. So I applied to Selwyn because they had the lowest number of applicants per place that year. Of course, they remarked very early on in the interview how they'd had a record number of applicants that year. And we realized that this is how schoolboys up and down the country were doing exactly the same thing. So I went into the pool, got pulled out by Robinson, and the rest for me personally was history. And I had a fantastic 3 years. My daughter, when she saw this slide as I was putting it together, she said, why have I just chosen these 6 photos? Well, it's very easy. They're about the only 6 photos I have from 3 years at Cambridge, because without smartphones, as we've got now, it would appear that there's virtually no documentation of my 3 years. But yesterday I was taking my son to Bristol University for his first day. We stopped at a service station, and outside there was a very big sign with a quote from Benjamin Disraeli saying that he had seen far more than he remembered, and he remembered far more than he'd seen. And I think that pretty much sums up my 3 years. I'm very happy with my very rose-tinted recollections of it. I do, however, feel a little bit of a sense that some of these photos I was almost taunting my future self with all that hair, which people I've only known for the last few years find it remarkable to imagine that I had. These were the medics, very blurry, but this was the Robinson Medical Garden Party that one of my photos was from. One notable absence, someone I'm still friendly with, Mohammed Nassim, he told me recently that from the day he started, he never wanted to be a doctor and never wanted to do medicine. And in fact, he gave it up the day he qualified, but it had a happy ending because he's now the CEO of a telecoms firm. But for amongst the rest of us, the remaining 9 of us, I counted 4 surgeons, 1 psychiatrist, 2 I've absolutely no idea what's become of them. I did have a good Google but couldn't find them, and one academic neurologist who's working in the University of Texas, and then me, who went on to become a paediatric allergist. So I trained in a specialty that suited me for a number of different reasons. I actually recall the very specific time I made that decision. I'd had an absolutely awful on-call. I always wanted to be a paediatrician, but I'd had a terrible night one night, and I went to a course the next day and saw an old friend there, and he said, you look terrible. He said, you're doing acute pediatrics, it's going to kill you. All those late nights, and you're still going to be doing them, in the hospital in the middle of the night into your 50s. So do pediatric allergy. I'm training in that at the moment. He said the on-calls are really easy. You don't get bothered. Well, that sounds good. He actually is now a general pediatrician and still up at night at Pembury Hospital, and I became a pediatric allergist. But I got really lucky because at the end of my training, the first consultant post I was able to get, Guy's and St Thomas's, it was a brand new service. They didn't have any pediatric allergy there, but they wanted some. And so they took on myself and 2 colleagues, both of whom were academics rather than clinicians. And I had the role of being clinical lead right from the start, alongside them setting up a big research program. And together we were able to build a service from the absolute bottom up, which was a really incredible journey. 10 years on, it's the largest service that we're aware of in paediatric allergy in Europe. We've had lots of very nice awards and recognition as a centre of excellence. But importantly, there's now about 100 of us employed in the service, seeing 7,000 or 8,000 children a year with a whole range of different allergic diseases. So a big bulk of my career has been focused on clinical and service development, which has been great. But alongside that, I've also had the opportunity to be involved in research. And whilst a lot of it was led by my colleagues, I also had the opportunity to do my doctorate alongside them. And it's always a challenge, isn't it, try and sum up your whole research career and all your publications in one slide. I'm going to try and do it, but essentially this is what it comes down to. We know there's a lot of people with food allergy around. It's something that was really uncommon. Certainly when I was growing up, there was one kid in my school who had a peanut allergy, and it was considered a really bizarre thing. Whereas now everybody knows people who have got food allergies, and we estimate it's probably about 5% of kids have got them. And the one that gets the most interest is peanut allergy, because it persists. It doesn't go away like milk and egg allergy do. And very sadly, it can cause very severe and even fatal reactions. And certainly it makes sense that if we can understand the mechanism of how people are becoming allergic, we've got a better chance of understanding how we can prevent it or possibly treat it. And that's what my doctoral thesis and a lot of my work has been around. And together with colleagues, we came up with what's called the dual allergen exposure hypothesis. So this idea of where you become allergic to something in the first place. One of the first things you learn at medical school about allergy is you can only react to something you've already had some sort of contact with before. But what was interesting about peanut allergy is it was when babies first ate peanut that they had their reaction, almost invariably, 80–90% of the time, which meant they must have come across it from somewhere else before they ate it for the first time. And people assumed that it must be in utero when the mums were still pregnant with them, that they were eating peanuts, or when they were being breastfed. But we knew that if you got cord blood from babies at birth and checked for allergic antibodies to peanut, there were none there. So these babies weren't born allergic. And we also know that a significant proportion of these babies were never breastfed. So it couldn't just be an issue of peanut crossing through breastfeeding. So we had the idea that it could be through the skin, and that we knew it was kids with eczema who typically got peanut allergy. We knew it was kids who had the most severe eczema that started earliest, that were most at risk. So we did a series of studies, both looking at the immunology, but also clinical pictures, and were able to demonstrate that it was all about exposure through the skin. So essentially babies with eczema, other people in the family eating things with peanut in, peanut butter, for example, kissing and touching the baby, that was the major risk factor for developing peanut allergy. And of course, the important bit about that is, well, what does that mean in terms of prevention strategies? Well, the one that we went down the route of was thinking about early introduction of peanut orally. So the idea of something that we already saw in parts of the world. So for example, in Israel, we knew that in infants, when from 4 months of age, so much earlier than we were recommending weaning in the UK, from 6 months, in Israel, almost all babies, the first weaning food they got at 4 months contained peanut, and we saw virtually no peanut allergy there. So we were able to confirm that if you've got Jewish kids born in London, Jewish kids born in Israel, looked at differences in their weaning habits and their likelihood of peanut allergy, and found that actually the same kid with the same genetic background born in London was 15 times more likely to get a peanut allergy than a kid of a similar genetic background who was brought up in Israel. And it seemed to relate directly to exposure to peanut early in the infant weaning diet. So the next thing, of course, is a randomized controlled prospective study to try and prove the point. We've got about 600 or 700 kids, all of whom had eczema and egg allergy, which is a big risk factor for getting peanut allergy. You've got a 20 or 30% chance of getting peanut allergy if you've got egg allergy. And then they were randomized to one of two groups. They either had to follow what was then the Department of Health advice, which is to avoid peanut in their diet, or they were deliberately introduced to peanut 3 times a week from 4 months of age in their diet. And by 5 years of age, there was an 80% reduction in the risk of peanut allergy in the ones who had that introduction of peanut early on. So really, really clear results. And I think, I think that the thing you can only ever hope for the most, not just getting these things published in good journals, but to actually find that what you've done has changed guidance globally. And that's exactly what that work did. And a couple of years ago in the US, we saw a clear change of public health recommendations to reflect the need of early introduction of peanut into the infant weaning diet, which is fantastic. Another large part of my career, though, was dedicated to trying to disseminate these messages. We found a lot of what we were doing was well known amongst ourselves, but just wasn't getting out there, even after things were published. And so one of a labour of love of mine was setting up something called the Allergy Academy to really get out there with tiny specialties, only a handful of allergists, yet so many people suffering and so many people who aren't specialists seeing those kids who have got those allergic diseases. So put a lot of effort into developing a large educational initiative, and that was recognised in 2018 when we were a finalist for the BMJ Awards. And another part of my career was other sort of leadership roles. So alongside my role in allergy, within my NHS trust, I became first the clinical director, and I'm now the commercial medical director for Guy's and St Thomas's. So essentially providing medical input into the whole range of commercial activities that the trust uses as a way of diversifying our income. All of the money goes directly into NHS care, but because we don't get enough central funding to do all the things that we'd like to do, we try and find other ways of doing it. So from private practice, commercial education, major industry partnerships, a whole raft of different ways of generating income safely and ethically that then supports NHS care. And then alongside that, at the moment, I'm also the first paediatrician to be the president of the British Society of Allergy and Clinical Immunology. And very much as Wendy alluded to earlier as well, because of these different roles, and especially being in a small specialty, that brings a lot of media activity as well. And that's been a really interesting part of what I've been doing. I've had some really fantastic opportunities to travel the world, meet some very interesting patients, some very interesting people. For me, my pinnacle was at the central picture there, getting the William Frankland Award for the contribution to the specialty. William Frankland sadly passed away this just earlier this year, age 108. He was 103 when I got that award, and remarkably, and then he retired from clinical practice about 3 years earlier. So certainly something of an inspiration. So the other thing we were asked to think about was the future of medicine, and certainly from an allergist perspective, whilst there's some really exciting work going on still around allergy prevention, the introduction of biologics, monoclonal antibody medicines is having a really big impact as well. But I think the real focus for me, bringing together my research interests and clinical interests as well, has been around food allergy, this enormous increase we're still seeing in the numbers of kids pitching up in A&E with severe reactions. And thankfully, fatal reactions are relatively rare, but it is not rare at all now to have small children showing up with really nasty reactions. And still, even over the last 5 years, we've seen a significant increase in the numbers of these here, Europe, US, Australia, everybody reporting the same thing. And for us, the area that I think is really changing things is food desensitization. So the idea that we can introduce very small but slowly increasing amounts of the food that you're allergic to into your diet as a way of promoting the production of blocking antibodies that get in the way of the allergic response, to allow you not necessarily to eat that food freely, but at least protect you from the majority of accidental reactions which happen to relatively small amounts of allergen. And I think this is a classic medical story. These are, you may notice on these stories from BBC Online, The Telegraph, these are 2009, 2010, and say very clearly at the top, cure for peanut allergies within 3 years. Here's a colleague of mine being quoted in 2017 about essentially the same studies, or just the continuation of them, saying this is going to be along in 3 years, still 7 years later. But delighted to say that in 2020, we've already had, early this year, for the first time, a peanut desensitization product in the US has been licensed. I was lucky enough to be involved in some of the studies that help support that. In fact, at Cambridge as well, there's another similar product being worked on, and that Adam Brooks offers peanut desensitization now. So this is becoming a reality, but that's just for peanut. There's a whole raft of other allergies that we need to sort out as well. And I think really for me, what the next 5 to 10 years looks like is developing this from its infancy to something that has a real impact in patients' lives. Thank you. John, I'm going to pass over to you.
Thanks very much, Adam. I'll just share my screen as well. And thanks everyone for joining. So I came up to Robinson in 1980. So we were the first year to occupy the new college building, 150 of us. And it was great to be in a newly built place. I say newly built, but actually that's what we arrived to. So while our rooms and the canteen were ready, the chapel, the college hall, and much more worryingly, the college bar were not actually yet finished. It didn't bother us at all, though. There was a lot of excitement about being part of a new college. Now, Freshers' Week is a very organised affair, but back then there were no clubs, no societies, so we just formed our own. If you found anyone else who was interested in what you were, you could form a club. And I remember being really proud and delighted to be selected for the men's rowing 8. Even though I'd never rowed before. And it was only later I discovered there were only 8 people interested in rowing. Like Adam, I have very few photos. It was even worse in 1980. You had to carry a camera, which really wasn't cool at all. And some photos I found I can't really show, but here's one of us in the rowing 8. That is me. I have to say our coordination in dress far outweighed our coordination on the river, but we were still very proud to win our oars in our first year. I remember us being well behaved and following all the rules, but the photos I found suggested we didn't always obey everything. And like Wendy, I remember happy days of croquet on Thorny Creek lawn, Mayballs, summer garden parties, and the rest. And academically, it was also very fruitful. We had a great admissions tutor in Alan Sharp, and his incisive sense of humour. Tutor of Chris Hughes, who's calm and compassionate. And my own director of studies, Herman Berrios, a neuropsychiatrist who's still an emeritus fellow. And I probably shouldn't use these words at a time like this, but the enthusiasm he had for his subject really was infectious. And I think rubbed off on me, and probably in no small part is why I've ended up being a psychiatrist. Now, I apologise for this picture, but in those days there was an event called the Birdman Leap, and it was a large, well-organised event, and the challenge was to fly from Silver Street Bridge to the Weir. And if you got there, you won a barrel of beer. And it was all very safe. There were police divers in the river to pull us out if there were any problems. And a few of us couldn't help entering as the dolly birds to try and complete the challenge. I have to say we didn't get very far in terms of flying, and the water was very, very cold, but we did raise quite a bit of money for charity. There were serious events as well, and the big buzz when the Queen opened the college in 1981. I remember the huge security and sniffer dogs around the place beforehand. Fortunately, the only sinister thing they managed to sniff out was someone's old sports kit rather than anything else. And also remember it was cold at times as well. And we had a big freeze in 1981. And it's not often you get to cycle or walk on the Cam. This comes with a health warning though, please don't try this at home. So many happy memories of Robinson. I did part 2 in experimental psychology, and I think that got me even more interested in sort of psychological medicine. I then moved to Oxford for my clinical studies. I mean, most students did then, medical students, they moved either to London or Oxford. That's completely changed now, and nearly everyone stays in Cambridge. In fact, the medical school here had to increase its places a few years ago to make sure that every undergraduate could be accommodated in clinical medicine, because the fact that some people had to leave Cambridge was putting people off applying because they knew they might not be able to stay for 6 years. So there's been quite a change there. I then trained in psychiatry in London, and I think the reason I did that is during my medical training I came across these disorders, mental disorders like schizophrenia, depression, and cognitive disorders like Alzheimer's disease. And it seemed awful that there seemed to be very little that we knew about them and very little that could be done. And I thought there's a real need for a much greater effort, both in terms of clinical care and research. And so that's why I went initially into psychiatry, then into old age psychiatry. I went to Melbourne for 2 years where I became interested in brain imaging. And then I moved to Newcastle where I carried on doing longitudinal studies of brain imaging in people with dementia, and then moved back to Cambridge about 8 years ago, not only because it's a fantastic place to be in a fantastic academic environment, but the imaging equipment, which is one of my main interests, is really second to none in the world. We've got great magnetic resonance imaging and great functional imaging. And so my field, although I'm interested in many different aspects of mental health in older people, it's primarily dementia, which we know is a huge and growing problem. That's an umbrella term, of course. There are many different causes, the big 4 being Alzheimer's, frontotemporal, Lewy body and vascular. And the impact of COVID, well, it's been challenging. It's been difficult for people with dementia. Dementia is unfortunately a risk factor for COVID mortality. A lot of people in care homes, as you will know, have dementia, and that's been very high mortality in care homes. And social isolation has also had a big impact. So it's been a tough time. Our clinical services and our research studies had to stop for several months, although they're now getting going again. And if I have to pull out some sort of positives, I mean, there are 3 things I'd pull out really. One is the way the clinical teams, the academic teams pulled together in the face of this huge challenge was just tremendous. I've never seen anything like it, whether that's in the NHS delivering services or the university delivering teaching and research. So that was absolutely fantastic. The second is we've had to change the way we do things, both clinically and with research, a lot more in terms of remote assessment, remote delivery. And although that's been forced on us, I think there are some aspects of it that are good and some that are here to stay, and particularly in terms of delivering research studies. It does allow us to have broader reach, to maybe do things at greater scale, maybe at reduced costs. So I think some of that will last. And I think the third thing is a much greater realization of the close relationship between mental health and physical health. These are not two separate areas. They're actually very closely related. And your mental health is very dependent on your physical health and vice versa. And I think there's a much greater awareness of that. In terms of dementia, my interest has been in trying to elucidate mechanisms underpinning different dementias, and then obviously thinking about trying to get treatments and ultimately prevention. And so the two things I'm going to link my research into the future is really about disease modifying treatments and about prevention. So what we know about dementia, and I'm going to focus here on Alzheimer's, is that various things in our genetic makeup and exposure to the environment make us more likely to lay down abnormal proteins. That's amyloid and tau, and these seem to be toxic. They kill brain cells and cause dementia. And a lot of therapies at the moment are aimed at amyloid and tau, and there's some promising advances being made in terms of disease modification. But there are also other processes we now know that are important. And so a lot of my research has been in terms of looking at vascular factors and looking at inflammation. And I think inflammation is really interesting. We know that inflammation is a factor of many disorders, and we know that it's a good thing initially, but that it can get out of control. And sometimes the inflammation can end up being worse than the initial insult. And we're now getting increasing evidence that that might be the case in dementia. And so we've been looking at brain inflammation early on in the course of dementia and finding there is evidence of quite a substantial increase, and that it does seem to be associated with the cognitive decline and progression. So there's a lot of interest, not just from our group, from others around the world, and whether this might be a sort of tractable mechanism that you can intervene with that might stop the advance of dementia. And the other thing is thinking about prevention of dementia. So we're also doing studies where we're looking at people at risk of future dementia. This is one, the Prevent Dementia study, where we're looking at a cohort of people aged 40 to 59 in midlife, and we're looking at various sorts of brain imaging. I won't go through the details, but we're looking at their structure, amyloid, tau, doing some sophisticated cognitive tests. And this study and others around the world are telling us that really dementia is not just a disease of late life. We used to think it developed when you got old. Actually, the process underpinning it goes on for 20 or 30 or more years. So while that might be a little bit depressing for people of my sort of age, that there might be things going on, it also allows a long period of time during which we might be able to do something to intervene and to do something to prevent dementia, which is ultimately what we want with all illnesses. And there are things that we know that seem to be risk factors for dementia. So there's some things we can't perhaps do an awful lot about: old age, genetic makeup, sex, but there are modifiable risk factors, and more are being described. These are 12 that the Lancet Commission thought there was sufficient evidence to say we don't yet know that modifying all of these will actually prevent dementia. And public health messages at the moment are really about these in red here. So keeping busy, avoiding social isolation, don't smoke, lose weight, be more physically active, limit your alcohol consumption, and look after your vascular risk. So these are great public health messages. That's what we have at the moment. But I think the future is probably bringing that down to the individual level, as we have done for things like cardiovascular disease, because maybe people aren't so interested about just doing all these things. It's what would lower your particular risk of dementia, given your genetic makeup, your age and your risk factors. And I think that kind of personalized approach of looking at individual risk is something that we're going to see much more in the future. And will be a big advance, I think. And that combined with disease modifying therapies, which I think is a realistic prospect of in the next sort of 5 years or so, will really make big advances, I hope anyway, in the future of my field. So with that, Judith, I'll stop and hand back to you.
Thank you very much, and many, many thanks to all three of you for excellent presentations, really interesting, fascinating, different fields and the slightly different ways in which you approach things. We've got some questions coming in, but before we start with those, perhaps if we touch briefly upon the field of medical education, how would you see how we can encourage medical practitioners training and medical education to go to address the needs of a very diverse type of population, diverse type of public that we're dealing with at the moment? And I'm leaving it open to any of you, perhaps if you'd like to pick up that thread in terms of what do we do with the medical students of today and with the young practitioners of today. Right, Wendy, would you like to start?
It's interesting, isn't it? I mean, in the time that I've been teaching medical students, there is quite a trend of change, actually, in the way that teaching is being done. And whilst I see some huge advantages, so for example, there's a huge instigation at the moment of things like team-based learning, problem solving, even for our first years. And I do find that incredibly exciting to teach, and we get very interesting ideas coming back from them. I would just perhaps say a word of caution that with so much knowledge available today, we do put huge demands, I think, on our young people to gain their own knowledge as background for that. And I think the challenge for medical education and all university education going forwards, but I think it's particularly pertinent here, is getting the balance right between what we expect them to be able to teach themselves, if you like, from the huge amount of information that's out there, and we direct them in that place, but it's rather sort of guided learning, versus what we want to take more control over the teaching of. I don't know whether that's sort of come across as clearly as I'm trying to say, but there's a huge shift, I think, in the way that we teach at university level at the moment, using all the online resource, and particularly this year, because we're being forced to give much more of a blended offering than we have done normally. But I am concerned that we've got to get it right. We've got to get that balance right between the one-to-ones, the group teaching and the larger class teaching going forwards to make it a good experience for them.
Any other views?
Yeah, I mean, I'd echo that. I mean, I think this is a time of real change. And I do worry a little bit on a number of levels. Firstly, when I speak to colleagues, nearly all of them tell them that they tell their children not to go into medicine, which I think is such a shame. I feel that I'm one of the few people I know that I work with that takes the opposite view. I think it's a fantastic career. But then recently I had we had a school reunion, and I met the 12 other kids that I left Haberdashers' Ascot Elstree in 1990 with, or had contact with. There's only one other who still works in the NHS along with me, which I found absolutely remarkable given the number of them. Most of them are still doing something medical, but it's either in the private sector or overseas or elsewhere. And I think all of this together, there's just an environment at the moment which just isn't inspiring our medical students in the way that we'd like to. And ironically, there's no doubt their hours are dramatically better than anything that we would have gone through as junior doctors, yet their experience is not as good. Despite what were barbaric working conditions when you think back to it. You know, we'd go to work on a Friday morning and leave Monday afternoon. It was madness. But actually, I've got nothing but fond memories for that period. So something isn't quite right in that experience. But I think, as Wendy's alluded to, it's certainly changing. And I'm very much hoping, I'm not an educationalist, and I hope that we come up with something a little bit better that inspires our students to want to stay in what is a fantastic profession.
And I'd simply add, I think the focus on core skills rather than facts, that's been a big transition over the last 30, 40 years. And that's really important because they're then generalizable to other situations in a way that facts just aren't. And the other, I think, is exposure. So exposure to greater diversity, and we need to do more of that with our students, and that has been happening. And I do think that, for example, the online other materials really allow that opportunity to ensure that students do get that very diverse experience and exposure, which hitherto has traditionally been very, very limited in all sorts of ways.
Thank you. We've got one question here, which is, how does the panel feel have been the most successful ways of bridging accurate medical knowledge across to the public? Shall we start in reverse order?
So it's a challenge. It's clearly a difficult thing to do. I think communication is key. The two different ways of approaching it, I think. So there's clearly some of the national guidance is really important as a framework. So things like the National Institute for Clinical Excellence, the sort of work that they do is sort of incredibly important. But then it could just sort of sit there and not be used and disseminated. So you need things in media. You need those of us here going on the media to disseminate it. But actually, you know, when I think of some of the big things that have come back to me in terms of what patients have said in the clinic, it's when the person on EastEnders gets dementia. That really brings it home to people because they recognise that, because most of us in our families have had somebody who's experienced that. So I think that getting it in very, very broadly is sort of incredibly important as well. And having champions, so the people who are brave enough to come out and say, you know, I suffered from this, that, or whatever, particularly if they have a large following, they're big public figures. That is also very, very important.
Madam, you and Wendy have talked about communication, so.
Yeah, and I'll really echo what John said. I mean, I remember in 2011, I was involved with a NICE guideline for the first time on the development group. And we talked about our concerns that once we put the guideline out and it recommended that there were going to be a need for more referrals for kids with food allergies, that we were going to create this absolute tsunami effect on allergy services that we were completely unprepared to deal with. And the guy from NICE sort of chuckled a little bit and he said, I really don't think that many people are going to read it. And within about 6 months of it coming out, giving talks to large groups of GPs and asking them how many of them had seen this NICE guidance, and 3 or 4 hands going up in the room. It has to be much wider and much more multi-channel than that. You need things coming from all different angles to affect change. We know if you go on the telly and it's got to be the right sort of thing, you go on breakfast TV, an enormous number of people see it and it can have real impact. But then humanizing the story, certainly within allergy, I think our understanding of this really happened a couple of years ago when there was a very sad and tragic death of a young lady called Natasha Laperouse. But the way that her family responded to that and the way that they harnessed the media, that within just a couple of years, that not only massively raised the profile of anaphylaxis, but that actually achieved a genuine change in the law to make things better for people with the same problem. Just goes to show that when you get it right, you can affect genuine change pretty quickly, but you've got to be very smart about how you do it.
Anything further, Wendy? Or
well, I suppose my contribution here would be to sing the praises of places like the Science Media Centre, who work incredibly hard to make sure that scientists are not scared or put off from talking to the media by sort of helping both sides of the party come to a sort of friendly interaction. And I think that, we need to encourage people at all levels who've got something scientific to contribute to say it out there. The other thing I sort of think going forwards in terms of education is that I do see that my own children have become au fait with the concepts of molecular biology, DNA, etc., obviously at a much earlier age than any of us did. But it is important, I think, that to make sure that carries on. Really good science education from the word go, I think is ultimately the way to get the public to really grasp new medical concepts, new scientific advances, and not be afraid of those.
Can I just ask one question, which has intrigued me for a while? How do you all see the way that we're moving more into digital type of medicine? I mean, John, you touched upon that, but it's slightly worrying when we see people complaining and commenting on the way general practice seems to have moved away from the face-to-face contact with the patient onto digital consultations. Do you have any views on that?
Well, I think there are advantages and disadvantages. It's not the answer to everything. That's true. And I'm a great believer in, as a psychiatrist, in the face-to-face contact. They're just things you cannot do in a video consult. They're things you can't see. They're things you really can't pick up. And as I mentioned when I spoke, this has sort of been forced on us in a way, although we have had some online, for example, cognitive behavioural therapy that's been widely rolled out for some time. So I think it's a case of not all or none. It's picking where it is appropriate and where it isn't. And, things like the telephone we have been using for follow-ups for some time. So that's not particularly new. We've just been pushed quite rapidly to do everything, which I think is too far. So we'll come back to a balance whereby some things will need face-to-face, and that's absolutely what we should do. But there might be others that can be dealt with quite appropriately remotely. And the same will be true of clinical trials. I mean, as I said, I'm interested in brain imaging. You cannot do brain imaging remotely. You have to bring people into a scanner. So those kind of studies we have to do face-to-face. But there are other things where we probably can do them remotely and allow more inclusivity, at least partly through doing that, because geographical constraints, etc., don't apply, for example.
Right, we've got a large number of questions coming in. I'm trying to address some of them. There's one here which says, as an aspiring medic, what global problems relating to medicine do you believe need the most attention, apart from COVID, of course? Adam, anybody wishing to reply to that?
No, I mean, I think at the end of the day, the biggest global issues remain clean water, vaccination, birth control, child nutrition, because of the enormous impacts. I'm completely biased, I'm a paediatrician, but just getting up breastfeeding rates is going to have way more of a health impact than anything I'm ever going to do as an allergist, that's for sure. It's still getting all of that right, and I'm still not sure that we teach global health in the way that maybe we should, or as well as we should. A little bit too much focus on, so I'm going to offend John here, on functional MRIs and the fancy stuff we all do, when actually the real determinants of health are those much more basic things. That's a personal view.
Thanks, Adam. Offence taken, but that's fine. I think that, I mean, I completely agree with that. I mean, we know that clean water and sanitation has done more for the world's health than anything else. But thinking ahead, and of course, we're all going to stick to our own topic, aren't we? But the big increase in dementia I talked about, not actually in high income countries. The real increase, the real wave is going to come in low and middle income countries. And so there's going to be a huge burden there that I think at the moment those countries are quite sort of unprepared for. So I think that's a big problem.
Wendy?
Well, I mean, as the infectious disease person here, I think Adam's already alluded to it, but in terms of global health, we still have malaria, TB, HIV, which are, a sentence for people in low and middle income countries struggling to get on. It's going to inhibit a child's progression, whilst those diseases which could be controlled are still out there. So I think, as we say, improved vaccines, improved information about how those diseases are spread, and getting the treatments to the people that need them can make a huge difference in those areas.
Okay, I think we've got time perhaps for one more question. There's one here which is asking the panel's view on how the current big data techniques will help the advancement in dementia prevention, in virus control and medicine in general, which is probably similar to what you've been talking about, but I don't know whether you want to address it.
Well, I think John sort of alluded a little bit to personalised medicine. I mean, for me, big data techniques that we would apply, things like understanding why some people are more genetically susceptible, for example, to poor outcomes following an infectious disease than others. And I see that as a way of kind of, in the future, knowing who needs to receive vital vaccines or different sorts of treatment than others. So I think for me, it's about personalized medicine coming forwards, making it and using artificial intelligence as well to sort of to process all of that. But the sort of genomics revolution alongside the big data, I think, is going to change forever the way that we think about who to treat and when and how.
So for dementia, I mean, there's a lot of interest in how our behaviours may change long before we develop cognitive symptoms and dementia. So that might be in where we visit and how often, or it might be to do with what we buy for our shopping, or even how many shops we go to and those sorts of things. So there's a lot of interest in the use of that kind of data to look at a population level as to whether we can predict some of these things. And the other use of data is in terms of using anonymised NHS records, so not breaching any kind of privacy or confidentiality, but looking in terms of that tremendous resource that we have in the NHS in terms of the data that's routinely collected along people's lifetimes that can tell us quite a lot about risk and protective factors and efficacy of treatments, which would take a long time in doing a clinical trial and cost sort of many millions of pounds, and that data is actually sitting there. So I think there's a lot more we could do in that regard as well.
Well, and all I'd add, just knowing that there's a lot of medical students in the audience, is that this really feels like once in a many generations coming together of all these different things. That is going to be a genuine revolution in medicine over the next 10, 20 years. And it's an enormously exciting time to be at the beginning of a career, because I think us old-timers are probably going to be broadly doing the same thing we're doing now by the time we finish our careers, but actually things are going to be quite profoundly different in 20 or 30 years. It'll be fantastic to have the chance to be involved in that.
Well, I think on that note, I'm afraid that we are going to have to come to an end. Our time is gone now. Many thanks to all three panelists for the presentations and for the discussion. Apologies to those that we have not been able to present your questions to the panel, but they will be up there, so we may have an opportunity to maybe type some answers to that. And also the presentations have been videoed, and I think they will be available for some time. So many thanks to all of you. Thanks to the audience for participating, and well, I wish you luck.
Thank you.
Thank you.